Good afternoon, everybody. Welcome to Scientia, and thank you for joining us on this beautiful and somewhat windy afternoon. I'm Jim Pomerantz, Professor of Psychology at Rice and Director of Scientia, and this is our fifth lecture of the academic year on a colloquium series we're calling simply Animals and Humans. In this series, we're examining our deep and complex relationships with the animal world, including how we interact with animals, how they shape our environment, how they shape our view of ourselves, how they affect our culture, how we benefit from animals and vice versa, and in particular today about the risks that are involved in that interchange, and in the instance of our topic today, the risks to human beings. So everything from malaria carried by insects, rabies carried by rodents and bats, plague, swine flu, all come to mind, mad cow disease, and bird flu. We've lived and we continue to live in a world where nasty things don't always stay put. They hop from location to location and from species to species, sometimes with horrible results. So our speaker today is one of the region's, in fact, one of the world's leading experts on a problem that's been capturing headlines and television news lead stories for some time, namely the threats that are posed to us of diseases transferring from animals to humans. So it'll be my pleasure in just a few moments to introduce Dr. C.J. Peters from UTMB Galveston. But before doing that, I wanted to thank our speakers from this past fall. They included Alistair Norcross from philosophy, Lisa Meffert from ecology and evolutionary biology, Jack Zamito from history, and lastly, Dr. Huda Zogby from Baylor College of Medicine right across the street from the Rice campus. If you missed any of these talks, all four of the videos are up on the Scientia website, so you can just go there and click on the links. Following CJ's talk today, we'll have three more talks scheduled for the spring, leading off with a talk from Carrie Wolf from Rice's English department on February 28th. That will be followed by our friend and colleague Tony Wright from University of Texas School of Medicine Department of Neurobiology and Anatomy. And that will be on March 21st. And then on April 10th, the evening of April 10th, we'll have the Bachner lecture. That will feature Irene Pepperberg and her amazing African gray parrot named Alex. And if you haven't checked out Alex, again, Alex will not be giving the lecture, we believe, but we'll learn wonderful things about what Irene Pepperberg is doing with that amazing bird. You don't want to miss that. Our format today will be the standard one. CJ will speak for 50 minutes or an hour, and that will be followed by questions and answers, and then a reception where you can talk further with him, and we can talk amongst ourselves right outside in the foyer. So now for the reason why we're here today, and that's to hear from our speaker, Dr. C.J. Peters. By way of background, I should say that Dr. Peters is the John Seeley Distinguished University Chair in Tropical and Emerging Virology at the University of Texas Medical Branch at Galveston, and I should mention that there are only two such university professorships at all of UTMB, and Dr. Peters holds one of those two. He's also Director of Biodefense in the Center for Biodefense and Emerging Infectious Diseases at that institution. His research interests include Rift Valley fever biology and vaccines, phlebovirus pathogenesis, SARS coronavirus, and monoclonal antibodies for therapy. Some of the emerging diseases that he's been closely involved with are the Rift Valley fever virus, the arenavirus hemorrhagic fevers, Ebola virus. If this is scaring the bejeebers out of you, just stay tuned. And the hantavirus pulmonary syndrome. He has more than 300 publications to his name. He's a Texas native, played a key role in tracking down the famous hantavirus that caused the mysterious deaths in New Mexico that most of us in this room probably remember pretty well. And he also battled a lethal Ebola outbreak among monkeys at a Reston, Virginia biomedical supply company, and this story was featured prominently in Richard Preston's bestseller book, The Hot Zone. Dr. Peters is also the co-author of a 1997 book entitled Virus Hunter, 30 Years of Battling Hot Viruses Around the World. If you think you work in a dangerous environment, you don't even come close to our speaker today. So Dr. Peters received his BA right here at Rice University in 1962 and then took his MD from Johns Hopkins in 1966. After being on Parkland's Hospital Internal Medicine House staff for 2 years up in Dallas, he became an NIAID research associate at the Middle America Research Unit in the Canal Zone. There he acquired an interest in tropical diseases, chronic virus infections, arenaviruses, and the ecological determinants of disease transmission. Then Peters went to UCSD, University of California at San Diego, and spent 3 years as a fellow at the Scripps Clinic and Research Foundation. From 77 to 91, Dr. Peters was at the U.S. Army Medical Research Institute for Infectious Diseases, where he began working as a research scientist and then became division director and deputy commander. While there, he worked on biothreats from emerging diseases, particularly hemorrhagic fevers. Then in 92, he moved to the Center for Disease Control, which of course we all know about. And then finally, in 2001, he moved to UTMB, where, as I mentioned, he's now both a university professor and a center director. So let me ask you, as always, if you have a cell phone, to please silence it so it doesn't go off as CJ is speaking to us. And let's tune in to CJ Peters, whose title is Emerging Virus Diseases from Animals to Humans. CJ Peters.
Thank you very much for that nice introduction. Well, I'm going to talk a little bit about where viruses come from, and I don't mean to go back to Thunderbolt and the primordial ooze or anything quite that basic, but in terms of where do they come into our lives and how do they get into our lives. To do that, I need to go back about 50,000 years. This is Herman Park at an earlier picnic site, and you can see this little band of hunter-gatherers doesn't have much opportunity to interact with large numbers of their own species. But they had viruses. They had primarily two kinds of viruses. They had viruses that they lived with that were chronic and latent, viruses like herpes and so on. They had viruses that came at them from nature, but they didn't have viruses like measles, polio, and so on. Those viruses came to us later. And this is an example of some of the data that led to that sort of thinking. If you'll notice that an island with less than 500,000 population doesn't circulate measles very much of the time. This is because the virus hits the population, runs through the population, immunizes them, and then dies out. So you need somewhere on the order of 500,000 people, maybe in one spot or in interconnected spots, to be able to actually sustain the transmission of these viruses. If you'll notice, Guam is an exception, but that's because there were so many U.S. Air Force planes flying through there on the way across the Pacific. So we basically come down to what I'm going to call three epidemiological strategies that human pathogens and non-pathogens use to get along. Herpes viruses such as varicella zoster, herpes simplex, CMV, the viruses that are only known as BK and JC, establish persistent and latent infections. They can activate themselves, they periodically are excreted, and they ensure that even in a small group of hunter-gatherers, they'll be maintained. These viruses often have a phylogenetic relationship to their host. For example, with the herpes viruses, there will be herpes viruses of monkeys and of mice and so on. And if you look at the phylogenetic structure of the different hosts, the viruses will resemble the host in terms of their phylogenetic structure. They've co-evolved, in other words. There are other viruses, the ones I talked about, who have emerged in the past and have been able to persist in humans by hopping from human to human continuously, always infecting new human hosts. That was the weakness that smallpox virus had that allowed us to be able to eradicate it, hopefully permanently. The other kinds of viruses we call zoonotic, from an anthropocentric point of view anyway. They infect non-human hosts primarily, and humans are only incidental in the in the cycle. Rabies is a good example. In spite of the fact that sometimes you'd like to bite somebody and give them rabies, it doesn't work very well. You get it from rabid animals. West Nile virus is an excellent example. If we weren't here to complain about West Nile virus wouldn't actually care at all. If you try to reconstruct historically what happened, you get the feeling that these viruses probably emerged about 5,000 years ago when irrigated agriculture emerged in the Middle East. And I remember hearing about that in my world history class in Hammond Hall, and I never thought I'd necessarily use it again, but I was glad to know about it. I'm glad to find a use for it. At that point, they were beginning to coalesce into cities of 100,000 or so. It just so happens that at that time, the progenitors of cattle were being domesticated, and the human measles virus and a virus of cattle called rinderpest virus seemed to be the two most closely related viruses in that family, and presumably had the same precursor, or perhaps even transferred from aurochs to humans. In any case, that was probably when these viruses began to come into humans and be transmitted among humans. Of course, those are the viruses that are the hardest to deal with and the hardest to control in many ways. There are a number of them. We've managed to control a lot of them: measles, rubella, polio. We still deal with the respiratory viruses, and we are still concerned about the emergences of new viruses. It's one thing for a zoonotic virus to emerge, like West Nile, and to have to deal with that, but it's a completely different principle if we're going to have a virus that's going to emerge with human-to-human transmission that we will have to deal with on a worldwide basis, day in and day out. And that's, I think, the major concern. And from what I've developed, I think you'd feel like that we probably would acquire that virus from animals and from nature. And this is, I've used old data in talks before, but this is ridiculous. 50 million years goes back pretty far. But this is to emphasize to you that with the separation of the continents, we began to evolve on each continent and subunits of continents that are carved up by mountains and climatic zones and so on. We began to evolve ecological systems. And while I can never get an ecologist to get interested in studying the viruses in a system, they are there and they're locked into those systems, and they need an integrated system to persist. And this is particularly true with vector-borne diseases like West Nile, for example. It's only an occasional virus that can escape because it can cross over to another vector, or it evolves, or something goes on to allow it to move to a new geographical area. And I show this as an example because it's kind of cute. This is what's called Wallace's line, or Huxley's line, or Weber's line, depending on whose ecological church you belong to. But on the west of this line, there are one set of viruses, and on the east, there's another set of viruses. For example, if you take the virus family flavivirus, Flaviviridae, the flavivirus over here is Japanese encephalitis virus. And it's sharply demarcated in its distribution, or it was. The flavivirus over here is, I'm blocked, the Australian version, which is, anyway, there's a different flavivirus over there. This flavivirus, Japanese encephalitis virus, has gradually crept across, has invaded New Guinea, and now is in the northern part of Australia, and is threatening to displace Murray Valley encephalitis virus, which is the virus in Australia. So these things move, and they tend to move slowly. They move with ecological change, and they worry people when they do that. And in 1992, there was a report issued by Josh Lederberg, Bob Shope, and Stan Oakes describing emerging infections and what the threats were, laying out some of the rules, and making dire prognostications. Another group met in 2003, and I was fortunate enough to be a member of that group, And our conclusion was that, yes, they were right, and that things were getting worse. And the list that they had made of factors that led to emergence was absolutely on target. The only thing that they had missed is that there's probably more interaction among these things, the sort of the perfect storm kind of thing, rather than any one gradient leading to emergence. One of the things that's interested me that in the virus world, microbial evolution does not usually drive emergence. Influenza is really the only exception where the actual evolution of the microbe drives the emergence. Usually it's the other way around. There's an opportunity for that microbe to occupy another niche, and it's able, through its rapid generation time, high mutation rate, and so on, to adapt to that particular setting. The viruses that I'm talking about, since they live in nature, are driven very strongly by environmental situations, and they are only secondarily driven by some of these other issues, except for travel, which of course gives them the mobility to find new places, new homes, new niches. The only thing up here that I think we have much control over is public health infrastructure. That's something that we can get a hold of, we can lobby for, and we can change. But things like behavioral variables and so on and so on are extremely difficult to modify. To make things worse, we are, as we all know, our own worst enemy. We are occupying a huge percentage of the Earth's surface. We're driving extinctions by our changing the ecological landscape. We're moving things around. We have biological invasions occurring regularly so that the ecological system changes as things go. And just an example of the upcoming crisis with surface freshwater, which may get to us before global warming does. And to make it worse, I used to have a bunch of slides for this, aircraft travel, number of rivers dammed, and all this sort of stuff. And they all looked the same. They were all exponential. So I just put this exponential in instead of having to go through all those slides. But it makes the point that things are getting worse, faster, or I don't know if worse, certainly more complicated and probably worse. So the paradigm that I usually use in my own mind when I think about this is that the viruses are genetically variable. And that sometimes is a problem, like influenza, but more often it just allows them to move into one of these multiple ecological niches, and the travel and transport provides the opportunity for them to evolve in ways that we might not find quite so congenial. I wasn't sure whether this was really the case or whether it was sort of a CNN effect, but the last 5 years I was at CDC, I just got a list here of what happened year by year. And does this say, your battery is fully charged, okay? Is that just the computer or me too? I'm not going to go through this in any detail, but many of these viruses are known, had been known at the time that they made their reappearance under some kind of pressure. But this virus and this virus, which are related, were new viruses to science. Not only were they new to science, but the genus they belonged to was a new genus. Well, here we are, and at the time these appeared in 1995, and we had known about paramyxoviruses for a very long time, measles, parainfluenza virus, and so on. And all of a sudden we've got a new genus on our hands. Ebola had been discovered in 76 and hadn't been seen since 79. Andes virus and these hantavirus pulmonary syndrome viruses were discovered in 93, and so on down the list. So things are happening that we didn't ever suspect. We had no idea even that the viruses that were going to cause them were there. So how could we prevent them? And that, I think, is a serious concern. A little case study. I want to give you 3 little case studies. And the first one that I will do is Ebola. This is based on a 1995 outbreak that occurred in Kikwit, Zaire, now Democratic Republic of the Congo. And Ebola outbreaks always get a lot of publicity because they usually have a very high case fatality rate. But it turns out that Ebola is a very funny virus. It exists in nature. We're not sure what the reservoir is. The available data as of a couple of months ago with some new discoveries suggests that bats may well be the reservoir, but that's still not proven. But it's not the problem that you might think it is. In spite of the very high case fatality rate, it's easily controlled. The way these epidemics start is someone becomes infected. They infect someone close to them, It sputters along, and then it gets in the hospital. And in an African hospital where there's no needle sterility, there's no syringe sterility, standards of hygiene are very poor, it explodes. And that's what happened here. These are, the yellow bars are cases in non-hospital workers, and this is when it got into Kikwit General Hospital. This was finally diagnosed, and we responded to this around the 16th of May. It was easy to diagnose. There was no big virological mystery, and really probably not any epidemiological mystery, but people just didn't tumble to it, and finally sent us some samples, and we readily isolated the virus. But Pierre Roland and Philippe Collin from our group moved in here on the 16th of May, and there were a handful of cases in people who were already infected and in the incubation period. And there was only one other case in a healthcare worker because they used proper mask, gown, glove, proper precautions that you would take in a modern hospital in the U.S. And the epidemic died out. You only get this from very close contact with people, usually with blood or secretions, although we did find out something that was interesting, I thought super scary. This is a histological section of the skin of one of these patients. The blue things that you see up here are the nuclei in the skin, in the upper layer of the skin. This red stuff is a stain that shows the virus and the viral antigen just under the skin, and in this case breaking out through the skin. So you are very close to Ebola when you're touching one of these patients, and we're sure that that's how some of the infections occurred. But basically, you're not close to Ebola if you're in Houston and it's in Africa, because it's not like smallpox or measles, which I show here as one case spreading throughout a population. But it's more like the bottom leg here, where one case really can't even renew itself in the general population under Western conditions. It's only in a hospital ward, and particularly an African hospital ward, that you can see some transmission, as you see in the upper circle. So Ebola's out there in nature. It is contagious from human to human, but not very, and it can be contained and controlled very readily. Next example I'd like to show you is SARS coronavirus. Now, it's called a coronavirus because of this crown around the outside of the virus. And this crown represents the molecules that it uses to attach itself to a cell and get inside. In 2003, note was taken of a disease that caused pulmonary infiltrates. You can see here on the CT scan better than you can on the X-ray. It was transmissible from person to person, had about a 10% mortality rate. Now, it appeared in 2003, but retrospectively it was operating in China in November of 2002. And one of the dirty little secrets in public health is that nobody tells you the truth if they don't want to get problems with tourism and so on and so on. But it was picked up in February on the rumor mill on the internet. China finally kicked in and admitted that they had, 300 or 400 cases here and there in southern China in Guangdong province. one of the physicians from Guangdong province, who knew only too well what was going on, went to Hong Kong and spent one night at a place that's referred to epidemiologically as Hotel M. Now, there is no A through N. It's the Metropole Hotel in Hong Kong, if you must know. Just like it's like there's no preparation A through G. There's only preparation H. Well, there's only Hotel M. And when some of these people left the hotel, they spread this disease to Vietnam. WHO issued a global alert on their own. I think a very brave thing for them to do, because you knew they would be criticized, and they had a chance of being criticized unjustly. I mean, they were justified in doing it, but if nothing had developed subsequently, they would have probably been pilloried. But they were right, and something did happen. It got into Canada, and we ended up having suspect cases in the U.S., but only 9 real cases in the U.S. And this is the Metropole Hotel. And if you ever go there, be sure and stay on the 8th floor and visit the room and so on, because it's all been cleaned up. But this one person, Physician A, who had come from Guangdong province, introduced this into a Hong Kong hospital. And also, Mr. H was hospitalized in Hong Kong. And you can just follow the worldwide distribution of this from one patient, one night, one hotel. Ireland, U.S., Germany, Singapore, Vietnam, Canada, a little bit in Thailand. This was, I think, a very impressive disease, and a whole lot scarier than all the Ebola that's happened. All infectious diseases that I'm familiar with have some kind of irregular pattern of sort of super spreaders. It's not any more prominent in anything than SARS. SARS is the most prominent, and you can see how one patient leads to chains of patients that go out in spreading fashion. This is an airplane flight where there were 120 people on the plane, and 22 of them caught SARS during the 3 hours they spent sharing a plane with the index case. Fortunately, the public health community responded promptly. It was possible to contain this disease through quarantine and a variety of other measures. The graph here shows you along the horizontal axis the days of onset before someone was quarantined, and the vertical axis is the average number of cases that occurred secondary to that patient. And you can see that by early quarantine, there's relatively little transmission, and late quarantine allows more abundant transmission. There were a total of approximately 1,200 cases in Hong Kong, and almost every case could be traced to another active case. People are usually symptomatic, so it was possible to quarantine a lot of those people fairly early or hospitalize them under controlled conditions fairly early. You can see 27% were hospital workers. This was a bad disease for doctors and nurses. It took a tremendous toll. 5% spread within hospitals. 19% was an unusual epidemic we don't have time to talk about. 16% was household contacts, and only 10% were like other. So it was pretty easy to focus on where this came from. And retrospective studies suggested that there were some things that helped a lot. Mask use in public, washing your hands with an alcohol-based preparation, and disinfecting your living quarters with bleach. This is some of the measures that were taken in this photograph. These photographs are from Taiwan, but similar things were done in China. Everybody in the affected area wore a mask when they went out. A lot of public places were closed down. You can see the only person here who's not wearing a mask is Sun Yat-sen. Everybody else is all bundled up at the various degrees of efficiency. Fashion gets involved. You can see this lady has a nice fashionable green mask with a nice fashionable shape. We saw a couple that I didn't get photographs of people with Walt Disney characters on their masks and so on and so on. It was, people adapt. Before you could go into a public space, a public building or a hospital, you had to have your temperature taken. This is a fellow who's going to the parking garage of a hospital, and this young lady is putting an ear thermometer in because that parking garage will allow him to go into the hospital, and he had to be checked. And, of course, everyone washed their hands before they went into one of these hospitals or other public places. There were dispensers of alcohol-based hand wash around. Every hospital modified itself because if a patient came into an emergency room, their emergency rooms are like ours. You sit there for a couple of hours before somebody gets to you, and you can cough on a lot of people in that period of time. So the emergency rooms were all modified. When you showed up at the emergency room like this young man with a broken leg, you still were checked. History and your temperature taken. And if you did not have a fever, you could go in and see the orthopedic surgeon. But if you did have a fever, you went over to a separately constructed, individually ventilated emergency room where you were cared for by personnel who were all wearing protective gear. Your X-ray was taken in a different X-ray room, and so on. This is not cheap, but it sure beats having to live with SARS. If you were hospitalized, when the nurse came to see you to take your temperature, this is what she looked like, and this was extremely stressful for patients and nurses. It was difficult to control this inside the hospitals, even with controlled ventilation. So generally there were either segregated SARS hospitals, or in this case, they emptied out the psychiatric hospital and turned it into a SARS hospital. This is not a trivial exercise in terms of effort, disruption, and money. And I know this is a fake from the internet, but I like it because it shows that it left a mark on everybody by the time this was controlled. The other thing that's an issue, and we've discussed repeatedly, is what's the surge capacity? How long could this have been kept up? And how about the materials? They were nearing the surge capacity on respiratory protective gear. And this, while this slide is a joke, it's still an element of truth to it because we wouldn't be able to deal with this on a large scale in the U.S. Most hospitals, including UTMB, have stocked respiratory protective gear. Now, where did this thing come from anyway? Well, this is a list of some people who were looking, made by some people who were looking for turtles that were endangered, and they went to a market in Guangzhou, and this is a food market. They have a very liberal idea about what's food, and they like variety in their diet. And this market is full of these critters of all shapes and sizes. They were cultured, and the guy who seemed to be the most likely to be implicated was the palm civet. It's a mongoose-like animal, and the information that was derived from these cultures and cultures of different viruses told a very interesting story. This is an unrooted tree of the SARS, I think this is the surface protein. Let's start here. The early phase of the epidemic looked like this, and you can see as you go into the middle phase of the epidemic, you're moving away phylogenetically, and finally in the last phase, when it was eradicated, you're phylogenetically most distant. In other words, this virus was evolving as it went from person to person. The next year, there were a handful of cases, and this is very interesting. These are the palm civet viruses, which lead you down this 202, 203 path. But 203 and 204 was not a pickup of the spreading epidemic. It was a new introduction. So there was a second introduction into the human population. So the point I'm trying to make is to go back to the idea about, yeah, these things evolve, become more efficiently transmitted, and do a lot of things, but the introduction is probably not driven by mutation. To make it even worse, it's probably not even a palm civet virus. It looks as though these bats that later yielded virus isolates are the original donor of the virus, and somehow it got into the market, into the palm civets. So this coronavirus, we know the coronavirus family, but it's a new coronavirus, and it's a new branch off the family tree. It doesn't look like the three groups of coronaviruses that we already know. So there's a whole group of coronaviruses out there circulating in God only knows what, bats or palm civets or whatever, that if we bring them together with us in the right way, may have an opportunity to cross over and may cause disease. I just want to point out a couple of the major differences here. We knew that in Ebola, mask, gown, and glove precautions were effective. We didn't know with SARS. It turns out they were, but they're very difficult. We knew that interhuman transmission was inefficient, and it turned out to be fairly efficient with SARS. Fortunately, it didn't spread to the general population. So we have, I think, a whole different order of magnitude of risk between Ebola and SARS. Now let's talk just for a minute before we finish up about influenza virus. This is a negative stain electron micrograph of influenza virus. And this little fringe out here, once again, are the molecules that it uses to dock to a cell, and it's called the hemagglutinin or the H protein. One of the reasons that we've always worried about influenza is the 1918 epidemic. These are deaths per 100,000 per year. And you can see we were getting a handle on things through the 19-teens, but when we hit 1918, influenza was able to adversely affect the mortality rate of the entire country. We don't know how many deaths there were worldwide. The estimates range up to 100 million. More Americans died here from influenza than died in World War I. It was a fairly unusual kind of influenza, too. This is a man who has this violaceous flush, and who the author of the article said these people almost always died, even though they weren't breathing hard at the time you saw them. And this guy is on his last legs with the blue lips and so on. But even when influenza isn't doing that, it's a problem. We have every year in the U.S. infections, hospitalizations, and deaths from influenza. The estimates vary from 20 to 36,000, and it's mainly in the elderly and the very young, although it can involve people at any age. There are many different influenza viruses. It's a lot more complicated than it is with, let's say, SARS. There are 15 hemagglutinins, this H, and then we also look at another molecule called the neuraminidase, and there are 9 neuraminases. So most of the influenza activity seems to center around wild waterfowl, ducks and geese. It's a GI infection for them. They don't have pulmonary involvement, and they don't get sick. They chronically excrete the virus, though, and you can swab a cloaca or even dip into a duck pond, and you can isolate these viruses. There are other viruses, though, that may not be strictly waterfowl. There's an equine virus that actually recently crossed over into dogs and has caused epidemics in greyhounds and racetracks. And there are a whole variety of these other viruses. In their swapping segments, these viruses have 8 gene segments, one of which codes for the H protein, one for the N protein, and the other 6 for other proteins. And they're swapping segments. If you take a single cell and infect it with 2 different viruses, you may get several different viruses out that are not like either progenitor. And we don't understand the rules for how those segments can reassort. And we don't understand the rules for what determines the pathogenicity or the transmissibility. So we're looking at this huge combination and mixing vessel of genetic material out there in nature. All over the world, you see some aquatic birds there reminding you that that's probably the central host. But every once in a while, one of these viruses gets a mutation that makes it highly lethal for chickens. This is a chicken barn that was full of healthy chickens in the morning, and you come back at night and they're 99+% dead. But this virus itself is not pathogenic for humans. The typical avian influenza virus doesn't bother humans. Let me just go back and pick that up again and show you what we've inferred anyway. This is an influenza virus. And here's the H protein on the outside, and the N protein is shown as these little spikes. And it has 8 RNA segments. So sometime a bird virus moved over to humans, and that caused the 1918 influenza outbreak. That virus went away. We have no idea where that virus went. But another virus, which was a human virus and not a bird virus, it had been circulating in humans, reassorted with an avian virus and resulted in a new virus, H2N2. Now, when you get a brand new virus with a new hemagglutinin on the outside, there's no immunity to it. And so it sweeps through the population and causes a pandemic. And that's what happened in 1957, and it happened again in 1968, and we're wondering what's going to happen in 2006 and later. Well, in 2003, we got the first hint, and it's been going on since then. An avian influenza virus has evolved that's highly virulent for humans. The typical thing that happens is a child on a farm is exposed to poultry. After a short incubation period, they get runny nose, respiratory tract symptoms, cough. They're hospitalized, and they're dead in 2 days. The case fatality rate on this is about 50%. Fortunately, it's not terribly transmissible from the chickens to humans, and it's not transmissible from human to human. So it stops. But it has succeeded in causing 148 cases with 79 deaths that we know about. Remember, countries don't like to talk about this. Come to Istanbul and enjoy the coffee and see the rugs, but you don't worry about holding your breath because we've only had 4 cases. But it's primarily Southeast Asia, but it's spreading. It's in Turkey and it's in Romania. You can see in the hatched area, the areas where there are cases, and it's moving westward. These are the flyways. We believe that this virus can be carried by the waterfowl and by migratory birds of other species. So we have the opportunity for this to spread worldwide. And even if it didn't affect humans, it's so highly pathogenic for chickens that it's a major economic impact. What are we going to do about it? Well, we have several ways to approach this virus that we can use to deal with it. There are antiviral drugs, and there are vaccines. The first antiviral drug against influenza was a drug called amantadine. I don't know if you're aware of the problem with antibiotics in animal feed resulting in antibiotic-resistant organisms emerging and being transmitted to humans. Well, apparently in China, they were putting amantadine in the chicken feed, and presumably as a consequence of that, all these viruses, these highly pathogenic H5N1 viruses, are resistant to amantadine and rimantadine. Oseltamivir, the trade name for it is Tamiflu. I think the oseltamivir is so you'll buy Tamiflu, but Tamiflu is such a silly name. It's like Debbie flu or Tamiflu, but the problem with Tamiflu is, although it has had trials in the other influenzas, it has not been obviously successful in the human cases it's been used in Asia. So we don't know if it's going to work. We know that in mice, if you infect mice, you need really high doses. We also know that resistance can emerge either in the laboratory, and there's been some evidence for resistance in nature, and there's just not enough. There's not enough to go around. If you have Tamiflu in your medicine cabinet, I would keep it there. I would. The other issue is zanamivir was developed as a neuraminidase inhibitor, and it's different from Tamiflu, but it was only formulated as an inhalation product, like an inhaled asthma medicine or something. It's even less available. However, the good news is resistance can emerge to zanamivir, but it's not cross-resistant with the oseltamivir-resistant strain. So it could be used in an oseltamivir-resistant strain. Vaccines, more bad news. The way we make influenza vaccines, every year, in the absence of a pandemic shift, there's a little bit of drift of the influenza virus away from the way it was the year before. So we have to change every year or two the virus that goes into the vaccine. And the shot that you've gotten, if you've been good and taken your flu shots, was made by examining during the summer all of the influenza strains that can be found, plus the ones from the year before, picking one or two to go in the vaccine, and then making the vaccine in a cycle that involves growing up flocks of special chickens that are clean and don't have latent viruses and so on, and making it in eggs. So you only get one shot, and you only get one cycle a year to make this stuff. Well, if a prototype H5 vaccine has been made, it takes 6 times as much antigen and 2 shots. So it's going to be a bear, logistically, to make it if we try to go that way, and no one is really cranking up in a major way. The other thing that is difficult to deal with is that there are actually 2 varieties of H5N1 that are closely related, but they don't cross-protect. The other vaccine that's now licensed is what's called a cold-adapted vaccine. It's a vaccine that was grown at low temperature for a long time. It lost its virulence for humans. It's being marketed as MedImmune or Flu Mist. MedImmune is the manufacturer. It's been marketed as Flu Mist. And you can use that mix and match business with the segments to put new H genes into it, and the backbone maintains the attenuated phenotype. No one's ever done this with H5. We assume that it'll be attenuated the way H1, 2, and 3 are. And so one of these has been made, and it's undergoing testing, but how long will it take to get it out there? So what that basically leaves us with is nothing. We have been less successful with mask, gown, and gloves and that sort of thing with influenza than we have with anything. We would undoubtedly try the same things we tried with SARS. But we may be faced with a major medical, economic, social problem such that we've not dealt with ever in the past. We don't know if this thing is going to become transmissible from human to human, if it will retain its virulence. But if it becomes human-to-human transmissible and retains even a fraction of its virulence, we're in for a real problem. And one of the things that I wanted to bring up is the way sometimes this stuff crosses over with bioterrorism. We are not prepared for bioterrorism, but we have done tabletop exercises to look at what to do with bioterrorism. Now, these are kind of imaginary exercises, but it makes everybody get in one room and try to figure out, what would I do if? And basically, these have all been very scary. In the first exercise, top off, nobody had any idea who was in charge. Is it the governor? Is it the mayor? You may remember something from Katrina along that line, too. And so, the feds may be in charge, but what are they going to do if there's a major influenza outbreak in 4 or 5 states? I assure you that CDC building is not, packed with epidemiologists that you just open the door and they come running out. It is, they're stretched just like everybody else. And there's no give in the healthcare system. We just don't have the beds. We've closed them down, et cetera. We have just-in-time delivery of masks and a whole variety of things. So I don't know. Top Off 1 taught us that. There's been some improvement in terms of who's in charge. Dark Winter was a smallpox exercise, and basically the lesson was that Congress should authorize smallpox vaccine for the entire population, and they did. And it brings up the question of what's going to happen when you can only vaccinate 10% of the people who need it, or 0% of the people who need it for influenza. Go on the website and read the CDC pandemic plan sometime and skim down to the appendix to see the priorities for who gets vaccinated. It turns out to be government authorities come up pretty high near the top because, after all, they've got to be taking care of things, right, and making sure everything works. Plague brought out some more problems, and then another smallpox epidemic scenario that was more on a global scale brought up the problems of what are you going to do with the vaccine have-nots. So I think we could have a very interesting time ahead. I think we can skip over that. So I'll just leave you with a quote from Sir Francis Bacon that I think is very applicable to emerging infections, because we learn something from every one of them, but every one of them is different. There's always, it comes at you from a different angle, and you require a different way to deal with it. Okay, thanks for your attention.
Thank you very much for a wonderful lecture, and thank you for telling us that we have an interesting future ahead of us. We have time for several questions, so if you have a question, if you could raise your hand, then Sydney Burris or I will get to you and ask you.
Being factored in, and the rate might actually be lower than 50%?
That's a very good question. The issue is, if 50% of the cases die, how many people were really infected, and did some of them just walk off with nothing more than a sniffle? Amazingly enough, the epidemiological studies that have been done around these are pretty crummy. Usually they actually bleed the family. That helps us have confidence that there's not a lot of transmission within the family. But it doesn't help us to say that in the next house someone became infected by a chicken and they didn't get sick at all. So I think the dogma is that there's very little subclinical disease, but the actual hard proof with good epidemiological studies is lacking. And I am totally amazed that the investigations haven't included this, but they haven't. The things that you, I mean, these are very rural areas where people live with their livestock. In Turkey, there were a couple of stories that came out. In one of the situations, they were playing with the heads from the dead chickens and throwing them at each other. The kids were, boys will be boys. And the saddest one was in Turkey. This one little girl had, loved her little chickens, and she was trying to nurse her chicken back to health, and she became infected. So there is close contact with these animals, but there's a lot of questions still. Yes, sir.
Assuming you have a reservation on a flight to New York tomorrow morning, very crowded, would you take any precautions? Would you wear a mask?
Not yet, but I guess I should have showed this slide. I think the odds are against us with flu compared to SARS, because with SARS, one of the things that was very helpful is they didn't get infectious until they actually had a fever and a cough. But influenza is infectious we don't know about this hypothesized H5N1 before people get ill. And there are a lot of subclinical cases and mild cases that shed some virus, not as much as the severe cases, but they shed some virus. So I would certainly, if I were in Ho Chi Minh City and there was some flu circulating around, I would get on a plane and go home, and I'd wear a mask on the way home. I don't think the risk is here yet.
Concerning the transmission of the bird flu, I've read that it's mostly migratory birds, but there was a recent article about the turkey epidemic that indicated it was more travel and trade related. Would you comment on that?
Okay. The question is does it get around mostly by migratory birds, or does it get around mostly by trade? And I think it's going, the available evidence is that it's going both ways. There have been some big jumps from China to Mongolia to Russia that are almost certainly migratory birds. And on the other hand, with all these agricultural diseases, most of the spread seems to be trade. One of the things that happens with trade also is you put a lot of birds together, so all you need is one infected bird. It's kind of like a daycare center or a college classroom or something like that. And you also have the problem that farmers don't like to have all their poultry slaughtered, even if they're recompensed for it. So there's a tendency for some guy to see some sick chickens in his flock, but he's got a hen that lays more eggs than all the rest of them put together. So he takes her over gives her to his cousin and says, take care of her. And of course, she's already infected. And you see that kind of spread a lot in agricultural related diseases, particularly in third world countries. We're so mechanized and so on that we're so, the agribusiness situation doesn't lend itself as much to that. These places are closed and new birds don't come in, and they're shielded from migratory birds, but I don't think it would keep it out here. But in an area where you have backyard farms and so on, if you have a farmer who has a small flock and a couple of them die and he tells the authorities what they do is slaughter all the chickens, and in Turkey, for example, they'll pay him market price, but he doesn't have any eggs for breakfast the next morning, and he's got to restart his flock. So it's not a very popular thing among the farming community at all. One of the things that I thought was a no-brainer was to vaccinate the chickens, and it turns out that just hasn't worked, and it's not clear to me whether it's a lack of effective vaccines or whether it really just isn't a practical way to get at it. But it hasn't really helped. This virus is a very scary virus. It kills people. It kills cats for some unknown reason. It kills house cats, and it's killed big cats in zoos. Waterfowl are normally resistant to any kind of pathological effect. They just carry it around. But some species of waterfowl are killed by this virus. So it's got an unusually high pathogenicity. Normally an influenza virus that you isolate and you want to work with in the laboratory, you find that you can't adapt it to mice. You have to serially passage it in mice and adapt it. These viruses kill mice first shot out of the bag. So it's a very different virus.
You mentioned that bats are being looked at as possible reservoirs for both SARS, Ebola, as well as other emerging diseases. What studies are currently being done to quantify this, and also what factors do you think are the main drivers for emergence from bats into the human population?
There are studies. There are a couple of groups who are systematically looking at bats, but they're doing it more on a survey basis. Let's get a few of these and a few of those and a few of the other. And that's not going to get us where you want to be as to why. The best speculation is that I have, there are two major races of bats, the megachiropterans and the microchiropterans. The megachiropterans are the fruit-eating bats, and they are, it turns out, there's a lot more of them per bat, and they're also good to eat, some people say. I've never eaten one. But they live in the areas where the trouble has been with Hendra and SARS virus from megachiropterans. They live in areas where their environment is being encroached on, their habitats being destroyed. The big Nipah outbreak in Malaysia occurred right after the year when we had the pall of smoke over Southeast Asia after the crop fires and so on, the drought and the burning off of the crops. And you can't help but think that maybe crowding and stress and so on may have resulted in movement of habitat into another area where pigs or horses, as the two viruses seem to jump to first, were exposed. The other, from the microchiropterans, it's just, I think that Ebola has been wherever its reservoir is, probably a microchiropteran, has been coming out periodically for a long time. It's just that you only see it occasionally when it gets into a hospital because the surveillance level is so poor. The SARS, we'll find out, but I'll bet you that it's a microchiropteran brought into the market, and there was an unusual year where the bats were stressed, or they had more contact with the civets, or there were more civets. People are trying to look at that, but it's not easy to get retrospective data.
Is there a seasonality factor with the spread of the virus?
We're talking influenza. In the case of ordinary influenza, as you know, it's primarily a fall and winter disease, although it continues to circulate all year. And there is a phenomenon where the virus spreads slowly and not very well noticed during the summer, and then when the winter comes, you have an epidemic in a new place that you didn't expect because there was no transmission at the end of the season the last year. When the pandemics happen, they don't appear to have any seasonality for the first year. They can start any time, and they can run for different lengths of time, presumably because with the typical epidemic year, you're dealing with a partially immune population and transmission is dampened and you need optimal weather. This is the speculation anyway. But with the pandemic year, you have brand new surface proteins on the virus and no pre-existing immunity in the population, and so it moves much more rapidly and much more readily.
Okay, thank you all for braving the elements to be with us today. Thank you, CJ, for an enlightening, if sobering talk. And please now let's all join CJ out in the foyer for some refreshments, which are certified to be safe. Don't forget, February 21st is Kerry Wolf, March 21st, Tony Wright, April 10th, Irene Pepperberg.